The cell cycle is a set order of events that occurs within the cell. It prepares a cell for division and DNA replication and results in two identical daughter cells. As a result of uncontrolled cell division, tumors can occur. This can take place in any organ or tissue. Within the cell cycle, three stages occur: interphase, mitosis, and cytokinesis.
Interphase is an active period in the life of a cell when many metabolic reactions occur, including protein synthesis, DNA replication, and an increase in the number of mitochondria and/or chloroplasts. At the interphase stage, the cell undergoes three parts: G1, S, and G2. Interphase prepares the cell for division before it undergoes mitosis.
In G1, protein synthesis and RNA synthesis occurs within the cell. This prepares the cell by creating proteins that will replicate DNA in the S phase. Other organelles and cytoplasmic components within the cell are duplicated.
In the S phase, DNA synthesis occurs and chromosomes are replicated into two sister chromatids.
In G2, the cell continues to prepare for division. Protein synthesis occurs further, and the cell checks to make sure all DNA and organelles are present.
Mitosis allows for growth, embryonic development, tissue repair, and asexual reproduction to occur. Mitosis in the cell cycle is the process in which two genetically identical daughter cells are duplicated.
During prophase, the chromatin within the nucleus begins to condense or coil, and the nucleolus disappears. Centrioles, with their fibers attached to the centromeres of each chromosome pair, begin moving to opposite sides of the cell and separate the DNA during mitosis. At the very end of prophase, the nuclear membrane of the cell disappears.
In metaphase, spindle fibers from the centrioles align the chromosomes in the middle of the cell. This is an essential piece of mitosis, as it ensures that when the chromosome pairs are separated, each new nucleus will receive one copy of each chromosome. Next, in anaphase, the chromosomes are separated and moved to opposite ends of the cell. Lastly, in telophase, the separated chromatids arrive at opposite ends of the cell, and new membranes form around them. The spindle fibers disappear, and the chromosomes begin to uncoil into chromatin.
Cytokinesis, however, is different in animal cells than plant cells. In animal cells, a fiber ring composed of protein pinches a cell into two daughter cells, each with one nucleus. In plant cells, the cell wall is so rigid that it requires a cell plate to be created. Vesicles made of cell wall material fuse together to cut the cell into two daughter cells.